All right. Hey everyone, my name is Joshua Brown. As y'all know, I'm in y'all biology class, and I've decided to actually do an exam guide review for chapters 10 and 11. They are very, I won't say they're similar, but they kind of correspond with each other. So I figured for those of y'all who can't make the study sessions that we do, I would go over it with y'all through this Zoom meeting, and hopefully you're able to watch this on your free time and review the exam guide yourselves. But I'm gonna go ahead and get started. So chapter 10, I have this on the study app. So the first question we're doing, we got what is cell division? What does it consist of? So I remember last time we were talking, we have cell division consists of karyokinesis and the cytoplasm. So with karyokinesis, karyo, the first part of karyokinesis is actually means it says DNA division, but it actually is talking about the chromosomes. So anytime you see the word karyo, think chromosomes. So karyotype is talking about the arrangement of chromosomes. Karyokinesis talking about the division of chromosomes. So karyokinesis falls into two categories. You got mitosis and meiosis, which is what we're getting ready to talk about now. So karyokinesis or DNA division, and you got, of course, cytokinesis, the cytoplasm division. Any questions on that?
No, it's actually, like now that I've, been to the study group, it's actually kind of straightforward now.
Yeah.
Yeah.
So mitosis and its roles. We were just talking about karyokinesis. So mitosis, I believe we were talking about this before. I'm not sure if it was the last study group or the previous one. But excuse me. You got growth and development. So during meiosis, what we're talking about next chapter, you have the sperm cell and you have the egg cell. And when you combine both of these together, but they both have 23 chromosomes, no pairs. 23 no pairs. So when you combine both of these together, they form a haploid cell. And once that haploid comes into existence, then you can have cell division. Now you can have mitosis again. So you're starting from one cell to what we are today. So of course, to get from one cell to us, very complex amount of cells, you have to have growth and development. So we're constantly and steadily going through mitosis to grow. And then of course you got wound repair, something that gets damaged or you cut or whatever it might be. Cells got to be go through mitosis to replace those cells. And then of course got asexual reproduction through bacteria and stuff. Some organisms actually go through asexual reproduction as well, but there's not very many of them. So moving on to the next one, we have the cell cycle, which I think you wanted to talk about some. So the cell cycle is divided up into two phases. You got the interphase, and you have the mitosis phase, or the dividing phase, I should say, because the cell cycle is also in meiosis as well. So the first phase, when that cell first come into existence and becomes a daughter cell, is the G1 phase or the cell growth phase. And during that phase, the cell is going to be making what it needs to survive. So whether it be more proteins, more ribosomes, more organelles of any kind, it's going to be made in this G1 phase. And actually, I think it's going to get a little bit larger. And during G1, it's important to notice G1, a cell has 23 chromosome pairs. You know what I mean by pairs, Miss Jessica?
Um, you mean like there's no, what do you mean exactly?
Okay,
I don't
so
know how to explain it.
I'm gonna put it on here so it can see me. Sorry if it, there we go. So what happens when I say 23 pairs, you have the like I said, the sperm and the egg, right? So when you got the new cell, the cell has 23 chromosomes from the sperm, and then 23 chromosomes from the egg. That's my dog.
I thought that was a person at first.
King, hush, hush. So, so you have 23 chromosomes from the sperm, and 23 chromosomes from the egg. And these chromosomes are going to pretty much pair together. So what I mean by that is you have a set of chromosomes, the father and the female, or the father and the mother chromosome, and they're both going to code for the same thing. So whether it be for eye color or hair color, it's going to be one chromosome. Sorry. Gosh. It's going to be two chromosomes, but they're essentially a pair. So you have 46 chromosomes total, but 23 are from the father and 23 are from the mother. And they exist as, here, I'll show you on a thing here.
Now, is it 23 pairs or 23 sets?
23 sets.
All right. I want to be sure that I have that.
Wait, what are you asking?
Oh, you had said the first phase is the G1 phase, and in this phase, the cell has 23 pairs of chromosomes. I was wondering if that was right, or is it 23 sets?
So,
because I want to be sure I have that right in my notes.
I get what you're saying. Well, they're referred to as a pair. So, in other words, you see over. Can you see over here on the light on the left on my notes here?
Yeah.
So think of this as the left side here as the father's side, and the right side over here as the mother's side. So this chromosome here comes from the father, this one here from the mother, and that there's like a pair. It's almost like they code for the same thing. And this is very important in meiosis, not so much mitosis, but it's very important in meiosis. We all have 46 chromosomes regardless, but these two chromosomes here at the top, they code for the same thing. You got that? And then these two chromosomes here, they code for the same thing. And these chromosomes here, they code for the same thing. So you have one from the father and one from the mother. You got that? That makes sense? Okay. So you have 23 pairs of chromosomes. So I kept on going down like that. I'll go down till I hit 46 altogether, or 23 pairs. Okay, so what happens during S phase is I actually got a little ahead of myself. So it actually won't look like that. It won't look like that at all. So it'll look like this. Because these chromosomes have yet to go through replication. So this is probably more likely what they'll look like. So the top one is actually what chromosomes look like going through S phase. So bottom ones here, these are G1. This is what they're going to look like. All right. After the chromosomes replicate, then it's going to look like this X shape here. Because what's happening is they're replicating for mitosis or meiosis, whichever one. Whether when it comes to that S phase, both mitosis and meiosis, they will end up looking like this. You got that?
Yeah.
So yeah, this is common for both mitosis and meiosis. They go through that replication. It's almost like if you were to divide it in half or whatever, it's a mirror image. It's identical. So they meet right here at the centromere, and the whole right side is the exact same side as the left. Now, another important thing to note is, like if you look at the top here, if this is the father and this is the mother, they're all going through their own separate replication. So even though they're pairs, they're not the same. So this chromosome here, it replicates, and this chromosome here replicates, but they're not related. Like they're not the same at all. They're two different chromosomes. You got that?
Okay. Yeah.
Okay, then after the S phase, once all the chromosomes replicate, then you go through the G2 phase, which is another cell growth. And as we were talking about last time, this is where the chromosomes start to make what they need for mitosis. All right, so we were talking about the cell cycle, and then we had interphase. We just talked about all this, the 46 chromosomes. During S phase, they all replicate, and it makes 46 chromosome pairs. So there are two types of pairs. You got the during G1 phase, the 23 pairs, and they're talking, when they're saying pairs, they're talking about homologous pairs. And homologous means parent, one father, one mother. So that term, you'll get more into it in chapter 11, but homologous pairs means one father, one mother. Okay. So G1, you have the 23 homologous pairs. And in S phase, you have the 46 chromosome pairs, which is talking about replication. And then, of course, you have the G2, and then you, of course, have the dividing phase. So that was interphase. And then we have genomes, prokaryotes versus eukaryotes. So prokaryotes, they have one DNA and one DNA molecule, and it's organized into a chromosome. Now, eukaryotes have, of course, more. We have 46 chromosomes to be exact. So we have, I guess, 46 DNA molecules. So that was a very easy one. Moving on to chromosome structure. So I was saying that chromosome structures are organized by a DNA double helix. All right, so that's the smallest unit, and they're made, of course, the base pairs. So you, of course, have the AT and the CG. A always binds with T, C always binds with G, and they form up the ladder of the double helix of the DNA strands. I'm actually going to show a diagram here to help out with that. I love diagrams; they just help you see everything better, right?
Yeah.
So if you can look really closely at the top part here, you can see the different colors. This green, blue, red, yellow. Those are the ATCG I was talking about. So this structure, DNA, is organized in the shape of a double helix. All right. So from there, that DNA structure is going to be wrapped around these proteins called histones. And you can see it over here. This is that jumbled up mess. That's the protein. And the DNA is going to wrap, I believe, twice around that histone. And eventually, after combining a bunch of them, you're going to have what's called a nucleosome, and it's going to look like beads on a string. You can see it here. So these beads on the strings are called nucleosomes, and they are coiled into a chromatin fiber. And I believe we were showing it over here, what chromosomes look like. Well, actually, interesting enough, when you have during all of interphase, actually, when you are talking about a cell, remember the cheek cells that we did during our bio class?
Yeah.
That's what it's going to look like here. So even when you have cell division, you're not going to see the lines or you're not going to see that. You're going to see just that big old mass of tangled up of DNA. It's all in a bunch of this is just big mass of DNA. But once you start to get to the mitosis or the dividing phase, I should say, because it also happens in meiosis, that's when you start to see this X shape here, and that's because that chromatin fiber gets condensed and starts forming that shape, so it can replicate and divide easier. I should say divide easier. So that diploid versus haploid. So we've been talking about diploids most of this time. Diploids are any cell that has 46, in a human rather, has 46 chromosomes. So that when I was saying the 23 pairs, that's a diploid. It's also called a somatic cell. So we have somatic cells, you have sex cells, and once again, this is also chapter 11. That's why I wanted to go over this, because they really intertwine. The diploid is going to be any somatic cell that has the 23 pairs of chromosomes. All right, so when we talk about haploid, we're mainly talking about the process of meiosis. And when it comes to humans, there are other organisms like plants and stuff that have haploid cells as well, but right now we're talking about humans. So what are haploids? Haploids is when you divide that set. So in other words, for humans, I said it's 23 pairs or 46 total. Haploids is going to be 23 individuals, like individual chromosomes. There are no pairs, none at all. And they're mainly for sperm cells and egg cells. So those are the product. We'll talk more about those on the next chapter. But for now, just know that haploid is referring to half of a set of diploids. You got that?
Yes.
No, I could have clicked that. So diploid, like I said, a cell with two matched sets of chromosomes. Haploid, a cell with one set of, or I'd say half a set. So it's in other words, it's just 23 chromosomes. And the way to remember that diploid, that di, means double. So haploid, half, di, double half. You get that? And then let's go on to the next one, S phase. Am I moving too fast?
No.
Okay. In the S phase, are replicated chromosomes considered two chromosomes? So it's asking a question. In the S phase, are replicated chromosomes considered two chromosomes? What do you think?
No.
No. Yeah, they're considered one chromosome. In fact, the replicated side is called a sister chromatid, and they're considered chromatids until they split off. Now, this is what's really important. We're talking about mitosis now, but in meiosis, there's a point where they stop being called sister chromatids, and we'll get more into that later. Okay, so the S phase and the chromatids, what are sister chromatids? We were just talking about that. So that the replicated side is called a sister chromatids. They are identical. Stages of mitosis. Okay, so we were talking about this last class. You have the prophase, which is when the spindles start to form. In fact, let's go ahead and look at a diagram. So I like to see stuff. I don't know about you. So interphase, like I was saying before, this is what the nucleus is going to look like. As you can see here, you can't really see the chromosomes here. It's just a big old mass. It's just like we were seeing in the cheek cells. You saw what the nucleus was, but you couldn't see really inside the nucleus. But when mitosis happens, you'll start to see the spindles form here, and then you start to see the chromosomes start to form here. And this right here, this is a pre-metaphase, pro-metaphase, and I like to think of it as you got prophase and you got metaphase. So pro-metaphase is in between them. What happens is the spindle fibers start to attach to the chromosomes, or the kintochore in the chromosomes, and everything's starting to get ready. It's everything's starting to go into place. And then 3, this is called, I believe on the study guide, she calls it the dancing of chromosomes. It's when the chromosomes line up in the middle of the cell. That's the dance, because it's almost like a line. It's a line dance, really what it is. They're lining up in the middle of the cell, and then those sister chromatids and anaphase are split apart. They're pulled apart one to the other. And in the telophase, everything starts to go back to normal. You have in cytoplasm, you have in the cytokinesis, you have 2 cells forming, 2 daughter cells, identical daughter cells. We just talked about that. Stages of mitosis, we just talked about that. Went to cytokinesis. All right, cytokinesis in plants. So vesicles are transported to the middle dividing cell, forming a cell plate. So I actually have a diagram on that if you want to check that out. I'm not going to really go too much into that one there. Bacterial cytokinesis, they divide by binary fission. Well, we don't talk too much about that. Just know that their process is binary fission, not cytokinesis. All right, so cell checkpoints. This is something I did not get to talk to y'all about. Well, I talked to some of y'all. I think a lot of y'all had to go to the last study session. But yeah, we are definitely going to talk about this on here. Very important. In fact, I'm going to pull it up on here to kind of give y'all an idea of what's going on here. So what happens during cell regulation and the checkpoints? So for the cell checkpoints, we have 3. One's going to be in G1, one's going to be here in G2, and one's going to be here during mitosis. So when I say in G1, it's going to happen right around here. If you can see my cursor, it's going to happen right here before S phase. And the purpose of this checkpoint is pretty much going to be to ascertain whether the DNA is gone. In fact, it wants to make sure, because S phase, if you remember, S phase is going to be the replication of the DNA. So of course, for that DNA to be replicated, it wants to make sure that that DNA is fine. There's no errors, there's no problems with it. Okay, so after the cell DNA divide, the chromosomes divide, you're gonna go to G2, and the cell, of course, is gonna get ready for mitosis. Well, then the G2 checkpoint is gonna happen right around here, right before mitosis, right here in this area. And the purpose of that G2 checkpoint is pretty much gonna be to ascertain. Now, I'm gonna go right back to another diagram real quick. You can see here. So the G2 checkpoint happens right before mitosis. And the purpose of it is to check to make sure the chromosomes were duplicated properly. And then, of course, you have the last checkpoint, which happens right in the middle of mitosis. I wouldn't say, I think on the diagram it had it right in between mitosis and cytokinesis, but that's not necessarily right. It's going to happen, I want to say probably in between anaphase and metaphase. This is going to make sure that the attachment to each kintochore to a spindle fiber is assessed. In other words, it's going to make sure, it's going to make sure that each spindle fiber is attached to a kintochore. And the reason why it's doing that is you don't want a cell with one side having 25 chromosomes and the other side having 21. One side having 20 and the other side having 26. So what it's doing is it's making sure that each one is attached to a kintochore. So when anaphase happens and they start to pull apart, it's pulling the right amount of chromosomes. Okay, so that's the checkpoints. Then you have the promoting points, the regulators. So with each phase, I'm not sure if I want to go back to the diagram or not. I'll go back to the diagram. So during the G1 phase here, from G1 to S phase, how do we know when it's time to move to the next phase? Well, the answer is simple. You have proteins. As the cell, the life of the cell, remember we were saying that G1, you're making organelles that the cell needs to live. Well, among those organelles, you're going to be making proteins, and one protein in particular is called a cyclin protein. It's a cyclin E. So G1, during the G1 phase here, you're going to make a cyclin E protein. And as the cyclin E protein starts to be made more and more and more, eventually it's going to hit a tape-off point where it's going to say, hey, we have enough cyclin E proteins. And once it hits that level, what's going to happen is those cyclin E proteins are going to bind to these cyclin-dependent kinases, or CDKs. So here's a diagram of them here. The cyclin E is going to bind to these CDKs, or these cyclin-dependent kinases. And that's not it. From there, these kinases, when we said cyclin-dependent kinases, these kinases are going to come and bind to the CDK. And what they're going to do is they're going to phosphorylize the CDK, and it's going to activate it. So what's going to happen, it's going to start doing what it needs to do to promote the movement to the next phase, whether it be later on during, like, say, for example, during the cell growth G2 phase, whether it be start making stuff where it can start dividing. These proteins are going to do different things. I don't have an exact list of everything, but just know they're going to promote the movement to the next phase of the cell cycle. So then once we get to the S phase, it's going to be the same thing, that those cyclin E proteins that have been made, they're going to start to taper off. They're going to start to disappear and slowly fade away. And then you're going to start making another type of protein. That protein is going to be cyclin A. And think about it this way: they're proteins that promote the cell cycle. So that's where you get the cyclin from. So during the S phase, like I said, it's going to be cyclin A, and it's going to steadily increase until eventually, like before, it's going to reach a tape-off point. And when it reaches that tape-off point, it's going to be, hey, we have enough of these cyclin A proteins. Let's go ahead and move on to the G2 phase. And once again, those cyclin A proteins are going to bind to these cyclin-dependent kinases, the CDKs, and they're going to do the after being phosphorylized by another kinase, they're going to go through the next movement or next phase, start preparing for the G2. And then, of course, you have the last one, the G2 phase, and their cyclin is the cyclin B. And all this is in the diagrams and the checkpoints and regulators. I have it organized so it doesn't confuse you or mess y'all up. But this is going to happen the same way as the other phases. That cyclin B is going to continue being made until eventually it reaches a tape-off point, and they say, hey, we don't have enough cyclin B. Let's go ahead and start moving towards mitosis. And after each phase movement, whether it be from G1 to S, S to G2, G2 to mitosis, after these phases, the amount of those proteins being made is going to taper off until eventually there's no more of those proteins left. All right, so more on regulations, or regulators, I should say. I believe next will be the negative regulators. Yeah, negative versus positive. So these cyclins, as you were talking about, cyclin E, cyclin A, cyclin B, these are all positive regulators. What we would consider a negative regulator will be the P53, and they have a job in the whole regulation process of the cells. Their job, pull up another diagram, is, I would call it the cell police, and that's what we were calling it in the study session. So think of the way I came up with this. We call the police the 5-0. That's their that's what we call them, their terminology or nickname. So think of the P53 as the cell police. Well, P5, think of police, and 3, think of the number of checkpoints. That's how I thought of it. 3 being the number of checkpoints, so you have P53. P5 police, 3 number of checkpoints. And what they do is, remember I was telling you about the checkpoints, the individual ones, what they do. So say during the replication of chromosome DNA, we have some errors, like some stuff didn't get replicated right, or it was supposed to be an identical, sister chromatid. They have some variability, and stuff didn't just happen right. There's some errors. So what P53 is going to say, hey, this is not right. We need to fix it. And we're going to put the cell under arrest. It's called cell cycle arrest. And what happens is they're going to go through the process of trying to repair the DNA. And if it can, then it's going to restart this process all over again. But if it can't, then what's going to happen is P53 is going to send out a signal to initiate apoptosis or cell death. So P53, like I said, think of them as the police. They put, if something's wrong, they'll put the cell under arrest. They'll try to fix the problem. The problem can't be fixed. Program cell death, and that's the negative regulators. I believe that was all for chapter 10, but I do have some questions on here that I'm going to go over real quick. Oh, forgot cancer cell. So we were just talking about P53. So if something is wrong with P53, P53 is a protein, just like the cyclin proteins. So if something is wrong with P53, what's going to end up happening is P53 will put the cell in cell arrest. If nothing is wrong, that's its job. But if there's something wrong with P53, then cell arrest never happens. The problem never gets addressed. The problem never gets fixed. So in a sense, going back to the diagram, it was on there. I just forgot to do it. Going back to the diagram, if there's a problem with P53, the cell cycle continues. And as a result, you can end up with a cancerous cell. So damaged P53 is very bad. Okay, that's how cancer can come about. So 2 types of karyokinesis. Of course, we talked about mitosis and meiosis. We'll talk about more meiosis next chapter. If a cell has intention to divide, has 23 pairs of chromosomes. So this is a question. So I was talking about thinking critically. So biology is all about thinking critically. And I was like, well, a lot of the tests, she asks questions that require you to think critically. So I was like, let's come up with some questions on here. So these aren't actually questions from the study guide. These are questions that I put on here towards the end because I wanted to I wanted us to think more critically. So it says if a cell has intentions to divide and has 23 chromosomes, what phase is that cell currently in? So the question is if a cell has 23 chromosomes, 23 pairs. So they're talking about the 46 total, the 23 pairs. What phase is it in? So think about it. If it had 23 pairs, 46 chromosomes, that means the cells have yet to go through DNA replication. Because if it had DNA replication, then they would have 46 pairs of chromosomes. But here it says it has 23 pairs of chromosomes. So we're assuming that it has 46 chromosomes. So the answer is going to be the G1 phase. It says what protein is needed to promote advancement to the next phase. So this is a question that's kind of if it was on the test, it would be a 1-2 question. If you got the first one wrong, you won't get the second one wrong. But it's actually what protein is needed to promote advancement to the next phase. And if the answer was G1, then of course the protein needed to promote it would be cyclin E for the G1 phase, from G1 to S phase. All right, so the next question, considering what happens during mitosis, what cell component would be produced in the G2 phase to help in cell division? Another thinking critically question. So it's asking during the G2 phase, what would the cell make to help in mitosis? And our answers are ribosomes, microtubules, proteins, or 46 chromosome pairs. So the answer for this is going to be microtubules, B. So let's talk about the wrong answers, and then we'll talk about the right ones. And I say right ones because this problem has multiple right answers. And if you look at the test that we do sometimes, you're going to see that the problem has multiple right answers, but there's always a best answer. And for this one, microtubules is the best answer. So let's go wrong answers first. 46 chromosome pairs. Now, if you think about this, we're talking about the G2 phase here. This is completely wrong because this happens during the S phase. So yeah, this is not going to happen during the G2 phase. So this is completely wrong here. All right. Then we're going to go to the ones that are, kind of right. You got ribosomes and proteins. So yeah, the cell's going to make ribosomes, and of course they're going to make proteins. Why do I say of course? Because we just talked about the regulators of going from one phase to another. In order for the cell to go from the G2 phase to mitosis phase, it needs what protein? The cyclin B protein. So what's going to happen is in G2 phase is making that cyclin B protein till it gets to a certain level. But what's going to happen when it gets to that level? It's going to say, hey, it's time to stop making this protein and go to the mitosis phase. So that protein is going to help it get to the mitosis phase, but it's not going to help out in cell division. So remember, it's right, it's being made in the G2 phase, but at the same time, it's not helping out in cell division. So it's right, but it's not. The best answer for this is microtubules, because those microtubules are going to be in mitosis, playing an active part, pulling those chromosomes apart from each other. So that's why microtubules would be the best answer. Think about it. When you're taking a test, the best, not necessarily the right answer, the best answer. Make sure you read all the answers, find out which one's the best answer. And genome, this is some stuff that I was looking at, make sure I didn't miss. So talking about genome is all the genes that an individual cell has. And it is, I'm not sure if I talked about it or not, but if not, I'm going to talk about it now. And if I did talk about it, I'm going to talk about it again. So when you have a DNA, you of course have the DNA double helix, and a portion of that DNA double helix is going to code for a particular protein, and that is going to be called a gene. And we're going to talk about a lot on that in chapter 12 when we start talking about Mendel's experiments. But yeah, genes code for a particular protein. And from there, you're going to move from genes to I have it here. You can move from genes to chromosomes, which is of course the whole DNA module. And then you're going to move from chromosomes to the genome. And genome is all the DNA inside of a cell. So I didn't talk about it before. Here it is here. So once again, you got base pairs, and we were talking about that before. That's the ATCG. You got the genes, and they code for individual proteins. And that can be those are the traits. And like I said, we'll talk about more of that in chapter 12. And then of course you have the chromosomes that we're talking about now. And that's the one DNA molecule. And then you have the genomes, which is all the DNA in the cell. And I say all the DNA in the cell because there are more DNA than just inside the nucleus. Remember, if we talk about chapter 4, there's also DNA located in the mitochondria and animal cells, and there's also DNA in chloroplasts in plant cells. But yeah, we're talking about genome. We're talking about all of the DNA in the cell. And that is the exam guide for chapter 10. And I'll see y'all again on chapter 11.